Hello, my dear students. How are you all? I hope that you are also fine and you are not wasting your time and you are having great preparation on immune system. Yes, of course, for your exam and for yourself. However, today I welcome you to today's online class. I am Rokshanara Pervin, Senior Lecturer, Department of Biology, Milestone College. Today's topics are roles of vaccines in immune system, concepts of vaccination, and the roles of memory cells in immunity. However, those two parts like vaccination and memory cells are of different criteria and they have a vast issues to discuss. Well, what is vaccine? Vaccine is a substance that is introduced into the body to stimulate the body's immune response. It is given to prevent an infectious disease from developing and the person becoming ill. Vaccines are made from microbes that are dead or inactive so that they are unable to cause diseases. However, there are several aspects of vaccines you see. This is a special kind of medical preparation given to provide immunity from a disease. Vaccines use a variety of different substances ranging from dead microorganisms to genetically engineered antigens to defend the body against potentially harmful microorganisms. Effective vaccines change the immune system by promoting the development of antibodies that can quickly and effectively attack disease causing microorganisms, and when it enters the body they can easily finish them, preventing disease and development. Okay, there are some other aspects. Just have a look and have a screenshot. I have to go forth and to go details about the different sorts of vaccines. Well, now what are the types of vaccines? There are many types of vaccines on the basis of the different germs have ever been discovered and experienced. Okay, generally vaccines are of 5 categories. These are kind of alternative or something like the pathogens are cultured to inactivate them first. As a result, they lose the capacity to infect. For example, rabies, measles, polio, mumps, tuberculosis, etc. Okay, this killed vaccine. What is the killed vaccine? Killed vaccines are the vaccines which are inactivated totally, especially against the polio virus. When polio virus are inactivated, they or killed, they can act as the killed vaccine. These vaccines are produced from the dead organisms and forming vaccines as a liquid form. Example, influenza, typhoid. Okay, and toxoid vaccines are secreted by the bacteria which are inactivated to make toxoid vaccines. This technique is reversed for diseases in which the secreted toxins are the main cause of the illness. Scientists inactivate the toxin by using a diluted chemical solution called formalin. The resulting inactivated toxins or toxoid is harmless. Examples can include the tetanus and diphtheria vaccines. Okay, there are some subunit or purified vaccines or subunit which are formed from the purified antigens. So only the portions of the germs that cause an immune response to be used to create subunit vaccines. For example, a virus which causes the virus spikes or virus something like DNA or protein caps can cause a disease. Only these are collected to form the vaccines. These portions of the germs are called antigens. Subunit vaccines can contain from 1 to 20 antigens. Since only the specific, spatially these are necessary parts of the germs are used for this type of vaccines, the adverse events actually risk is lower. Examples include hepatitis B vaccine. What are the conjugated vaccines? There are two components are conjugated to form this vaccine. Usually a carrier which is protein in nature and the part of bacterial membrane is conjugated together. For example, the vaccine which is made from the Haemophilus influenzae type B, or you can see in the video that another example of the conjugated vaccines are as your book, they are mentioned only the influenza, and maybe it is used for the pneumonia. Okay, so there is another less important, but though you have to know, that surface chemical vaccine. These vaccines are produced from the surface chemicals of the microbes, like H-base AG antigen present in the capsule of the hepatitis B virus. This H-base silver antigen or G antigen isolated and produced vaccine through genetic engineering technology. And lastly but not the least is DNA vaccine. This is formed by the extraction of the portions of germs DNA, and make copies of it to create a DNA vaccine to make actually kind of recognition for the opsonization process. This type of vaccine would hopefully produce the better protection like live virus vaccines, but with even less likelihood of producing the diseases. There are currently some studies on DNA vaccines for mumps and herpes and influenza. We need to know the principles of the vaccines. The first principle is the vaccine consists of attenuated or killed microbes or their byproducts. For production of vaccines, we should not and we must not use any other chemicals which are not closely related to the germs. Okay, so the attenuated microbes, when used as a vaccine, they are able to multiply within the body after inoculation, means invasion, but do not produce any diseases. These toxoid vaccines could not produce any damage to the body, but they may help to produce antibodies. Immunity developed from vaccination do not last lifelong. It has some limits, but the lifespan may be extended by some booster doses. The booster doses can sustain for lifetime immunity. The fourth principle is the durability of the immunity sometimes depends on the ways of vaccine administration. For instance, vaccination gives instant permanent result when applying it through the some sort of alimentary canal in the case of intestinal infection. Okay, the immune response period is much longer than the incubation period. What is incubation period? Incubation period can be defined as the inactivated times or the duration where they are not feeding anything or invading anything. Hence, vaccination is not effective after the infection of the disease, with one exception of the hydrophobia. Actually, in case of hydrophobia or something like in Bengali, we know it is jolatonko by the by the bite of the cat, something like dogs which are also infected, right? This is kind of called rabies. But after happening this disease, we may have seen that lots of injections are pushed. Actually, not lots of injections, more than 7 to 10 injections surrounding the navels. Okay, then the incubation period is very long though. That doesn't mean that you are getting the bite yourself from the dogs, right? Anyways, so there are some national vaccination programs. Okay, you must know that at your book that the polio vaccine, DPT vaccine, BCG, etc. There are some notes at your book at page number 610 or something like 608. Maybe it's different at your book, but you have to finish this all. However, let's discuss about the roles of memory cells in immunity. What is the memory cell? They are the long-lived cells of the immune system that have previously encountered the specific antigen. You see this is the bacteria, right? This green colored bacteria, and this is the B cell, and in presence of this bacteria, they are forming the special sort of antibody, right, to kill the germ or kill the antigen from the bacteria. The red portion is the antigen, green is the bacteria which is releasing this antigen here, and the B cell is showing the antigen receptor by forming antibody. This is the antibody. Okay, so in the previous class you must know that how antibody is produced, right? Then the helper T cells scan them all, and this helper T cell produces the kind of plasma cells, another is memory cells. Two types of generations are produced. Clear? So the first step, the B cell finds an antigen which matches its receptors. Number 2, it waits until it is activated by the helper T cells. And number 3, then the B cell divides to produce plasma and memory cells. These memory cells reside in your body until according to their longevity, of course, maybe be extended up to lifetime. But these kind of memory cells help to kill the germs or inactivating the antigens without letting us know. For example, pox, right? When you are suffering from pox for first time, the antibodies are produced in your body. These antibodies are preventing lifelong from the invasion of the pox virus, right? You see, this plasma cell finally creates the antibodies and kills the bacteria, and that one looks delicious, right? This one, it is telling that this is delicious. Then this one, I remember this one, red alert. That means what? In the next time when this sort of the same bacteria invade in our body, these memory cells smiles like a kind of ghost, and it just knows that what kind of antigens are produced from here, from the previous experience, and they kill them directly without wasting time, right? This is very interesting though. Yeah, you see some diagrams of the helper T cells, cytotoxic T cells, and T regulatory T cells. So helper T cells produce CD4, and T cells receptors are bind over there, binds to MHC part 2, and cytotoxic T cells are actually producing the CD8 and to bind the T cell receptors, which is related to MHC1, and T regulatory cells are producing both types of antibodies, this CD4 and CD25, and it is the binding site of the T receptor cells. Well, there are some important aspects of the memory T cells. Have a look. Just focus on the third point. They are activated by small quantities of antigens and require less co-stimulation than do the naive and unactivated T cells. That means what? They are activated in a very small quantities of antigens, and they are triggered to be there to kill the antigens or inactivate, whether the number is small or large amount, doesn't matter. Okay, activated memory cells produce greater amounts of interleukins than do naive T cells when they are first activated. Okay, so comprise two subtypes: central memory T cells. These are important for MCQs though. TCM cells and effector memory T cells. Okay, just TCM cell means central memory cells, and TEM means the effective memory cells. Memory cells may be either CD4 or uneven CD8. Importance of memory cells in immunity. You already got to know that how they are important, right? The principal functions of B cells is to make antibodies against antigens, perform the role of antigen presenting cells or APCs, and eventually develop into memory B cells after activation by antigen interaction. On the other hand, T cells constitute 60 to 75% of blood lymphocytes. They can be distinguished from other lymphocytes by the presence of a T cell receptor. So it acts as the activated receptor on the cell surface. Another key feature of B cells and T cells includes the receptors it has in its surface. T cells recognize a linear sequence of amino acids, whereas B cells the spatial arrangement of proteins, that means one by one in an array in a linear form, and nucleic acids and polysaccharides or lipids. There are some functions of some cells. Have a look and have a screenshot. Plasma B cells and memory B cells, as I already told. Have a look. Yes, this is the funny image, of course, to make you easily understand how the antigens are disappeared. That the more the clone size, the more the clone size, the more the time is consumed. That means on the basis of the size of the memory cells, the time depends. So it's better to choose the smaller shaped memory cells, right? However, naive, that means just like stupid cells, or just get some sort of expansion, and when they die, they are diluted like this, and some are released, some get escaped as a memory cell, and they hide in somewhere safe place, and the other one just gets a copy. Means these four have the copy called tolerance. This just tolerance means they could get adopted. That means they are adoptotic cells, and this green are the memory, just precursors. So you need to understand. However, we got to finish the lecture today. Whatever, if you have any question, I have tried to put as more notes as possible, because you don't have enough resources to find this out. Just this, every page is important. You have to get screenshot and go through the details from the book or even internet. So here is the homework. Explain the roles of memory cells. You need to write it in a 4 mark standard section, standards or higher skilled section type. So thank you very much. Off to go.